I should explain what all of this means, I guess, before I even get started, so it makes some sense to you. More or less what I'm going to be talking about is this phenomenon of gene swapping between individuals. And this mostly, when I mean individuals, I mostly mean bacteria and other little single-cell things. So it's not like you getting some of my genes. It's single-cell organisms. So the question, I guess, it's become interesting because, one, we realize that it happens a lot. You may, of course, be aware from news reports that things like antibiotic resistance emerging in bugs in hospitals is something that spreads very quickly. And that kind of resistance is as a result of genes that enable bugs to be resistant to antibiotics entering into new bugs. So that this seems to be a rapid mechanism by which genes are spread, and those genes can impart new characteristics on those bacteria, for example. They can tolerate antibiotics, for instance. And from the point of view of people interested in the early evolution of life and in evolution in general, this topic has become quite interesting for a number of reasons, and I'll try and explain those as I go along. So just the first thing I'll show you is just then what I mean by gene swapping. So here's just a little picture. This is a very artistic rendering of a bacterial cell. So we have a cell here, and normally it will divide into two daughters, like this. And so each of the daughters will receive a copy of the genetic information, the DNA, and that's what we call that vertical inheritance. So that's sort of the same thing as what happens when we produce children. So that's the normal way of transferring information. But we also see this phenomenon here, which we call horizontal gene transfer, where genes can be transferred from one individual to an unrelated individual by a completely different set of mechanisms than this here. So there's no cell division. This is unrelated to that one, and it's picked up the genes from that individual.
Could you just define genes so that I not
Gene, for today's purposes, will just mean a stretch of DNA. So it'll be something that carries some information. It may have, and when a gene is, when I'm talking about horizontal transfer, I may be talking about one gene, or I may be talking about 100 genes. We see big chunks of DNA being transferred in very little chunks. And so the minimum that I'll be talking about would be one single gene, and that would then code for some, perhaps a protein that does something in a cell, like, for instance, resistance against an antibiotic.
But you can perhaps also say that it's never the entire DNA content of one organism that gets transferred in what you are talking about now.
Yeah, well, I'm not going to talk about all the DNA from one individual going into another. In principle, that can happen. You can do it in the lab, but it probably doesn't really happen in nature. So we're just talking about this process of swapping DNA rather than inheriting it from your parents, if you like.
What happens to the original part that was there before?
That's something I'll talk about, but in some cases, I guess the basic thing would be that in a successful gene swap, you received an extra gene. So if you had 100 genes and you picked up an extra one, you'd have 101 genes. Okay, so I'm going to talk about gene swapping because it's important for three sets of ideas. One is this tree of life, which I'm sure you're all now familiar with, the idea that all life on Earth is related by common descent. It's evolved from some common ancestor. And it's been possible to show that by looking at gene sequences. You take a bunch of gene sequences from different species, like humans and plants and yeasts that you use for brewing beer and bacteria and so on, and it turns out they all have the same version of a particular gene, and you can compare the similarities and differences in those genes to construct their relationships. And if one does that, one ends up with a tree, and that tells us something about who's related to who and in what way. Now the problem is that if the genes start swapping around, maybe the tree isn't very accurate any longer, because it might be following the genes, but it isn't necessarily following the organisms. So it's sort of like musical chairs, if you like. So I will address this point to some degree at least. Can the tree tell us something? Is this transfer such that we eliminate the tree? The other thing is then that it's become very popular to think of horizontal gene transfer as something that would have been a feature of the earliest systems, biological systems. And so if one reads some of the literature surrounding horizontal gene transfer, the implication is that this was just a default state in early systems. They were capable of transferring genes, and only later did they evolve some mechanisms to prevent genes being transferred. So initial transfer doesn't need to be explained under those kinds of models. The other alternative that hasn't really been discussed, but which I will discuss now, is that rather than saying it was a default state, perhaps horizontal gene transfer is the product of evolution. So I will try and make the case for that today. The third thing is then an idea that has, you may be familiar with this idea of Lamarck's model of the inheritance by acquiring new characteristics. And I will run you through that later on. But the reason it's come up is that in a recent essay in Nature by Carl Woese and a colleague, they made statements saying that effectively we think horizontal gene transfer is an example of Lamarckian evolution, and that Darwinian evolution here does not really apply. And I will, since it's been published in Nature, of course it will get a lot of attention, and I will tell you why I think this is a dangerous and incorrect idea. Okay, so if we start then, I'm going to be bad-mouthing Carl Woese quite a lot in this lecture, but it's not because I don't like the guy. So he has written a series of influential essays in the last, I guess over the last nearly a decade now, describing this greebly thing here, LUCA, which is shorthand for last universal common ancestor. So all you need to know about that is that if you build a tree and you say all life is related, the tree will have this sort of a branching pattern like this, and LUCA refers to the deepest point on that tree. Okay, so it's saying that if we took a simple example like humans, and then chimpanzee and gorilla, then this tree would be indicating that human and chimp have a common ancestor at this point here that has now gone extinct, and that the human and chimp lineage was once one single lineage rather than two separate, and they had a common ancestor back here with gorillas. So these ancestors have then given rise to different lineages. Okay, so this would be a common ancestor, and this would be a common ancestor, and if we took all of life, we could do the same thing with the whole diversity of life, and then we'd have a universal common ancestor. It's the last, we call it the last because there can be earlier universal common ancestors in principle, so hence that's why we call it LUCA. Now his concern is that the model that he's built is that well, the tree itself has come under a lot of fire, and I'm going to talk about that briefly. But to I guess say this in a nutshell, there was a lot of debate as to whose tree was correct, because people were producing different trees with different outcomes. So there was a lot of fuss. And as became clear that horizontal gene transfer may be playing a role in the inheritance of genes, that they're moving about, meaning that that might explain why your trees are different, then I guess it became more popular to talk about horizontal gene transfer very early. That's why it's messing up our trees, so we need to explain what's going on. So this is a little quote then from one of his papers, and he says here, any lineages involving barriers to the acquisition or expression of foreign genes, so any lineage that prevents genes from entering it, would be left behind in the evolutionary progression towards modern life. A lateral gene transfer, that's the same as horizontal gene transfer, of this kind and intensity would not only contribute significantly, but would also completely dominate the primitive evolutionary dynamic. So what he's basically saying then is that horizontal transfer was so prevalent that in fact it was more prevalent than this vertical inheritance from parent to offspring. In fact, he's saying that it would completely dominate it. So his model says then that genes could transfer freely between any two cells in some hypothetical system, so there will be complete genetic mixing, that was more important than this vertical inheritance. And his model essentially says that modern forms of cellular life, that's these three domains, the bacteria and the archaea, and then ourselves, the eukaryotes, they've all more or less come out of this cloud of gene swapping that happened very early on, and that may explain why they're so different. Now, so that's quite different from then our standard picture of a tree. You can have extinctions, and you also have events like this that lead to new lineages being formed, a single lineage and then two new lineages. So this is more or less how we traditionally viewed the evolution of life. There may have been earlier common ancestors, but the only ones that are left are these ones here shown in yellow, which produce a tree like this: eukaryotes, archaea, and bacteria. And all the other lineages have gone extinct. So that doesn't take horizontal transfer into account, specifically in that tree. And so what started to be debated perhaps around 7 or 8 years ago now were these sorts of models here. We knew there was a bit of transfer because we knew that, for example, all animals and plants and fungi have these things called mitochondria. They're your powerhouse of your cell. They're required for doing most of the key energy processes. And those were once upon a time free-living bacteria. They came into our lineage a long time ago. And we know that some of the genes have transferred. So these were horizontal transfers that we could accept. The same is true for chloroplasts that harvest energy from the sun. And they were once a form of bacteria, probably related to the cyanobacteria. So then, from this model, where we said, oh, we can accept that there can be some of these complicating features of the tree, sort of went to something a bit more like this, or even more like this. That the genes are swapping all over the place, and that the trees are not really trees, they're more networks. So they can, rather than having this sort of bifurcating structure, they can in principle close in on themselves like this, so that you would have a sort of a reticulate structure rather than a branching structure. And so these are sort of schematic versions of that. How much is that happening, and if that's happening, won't it ruin our ability to reconstruct a tree? The gene relationships between genes will not reflect the relationships between the cells, therefore using the genes in the extreme interpretation was that that would be a waste of time. It wouldn't tell us anything. The information was all gone. At the same time as this was happening, we were starting to get whole genome sequences, and that meant people were able to compare those sequences and try and work out, okay, if we take these different genomes, a collection of genes from a particular species, so this was a couple of the first two bacteria that were published, and you say, well, which genes do they have in common? How many genes are in common? You can put numbers on the common sequences, and this was just between two species here, and you had around 256 genes. As you added more genomes here, more different complete sets of genes from different species, the number went down. So it's probably the ones that seem to look like they produce the same tree might be somewhere in the order of 50 to 60, and that. What we can say about that number is it's too small to reconstruct an organism. It's only about, numbers-wise, it's about enough to make the machinery that produces proteins. In fact, most of the genes that seem to date back to the last universal common ancestor by using this type of approach, comparing the genes and seeing what's in common, are just required for making proteins. So there's not enough there in common to produce an entire cell. So that sort of underscored people's feeling that, whoops, it's probably the case that there is an awful lot of transfer. It's ruining our trees. I would say that, in fact, there's still some there that can be used. But the question still remains, what about the rest of it? So I'll come back to that perhaps at the very end. But what I want to focus on first now is this model of Carl Woser's. He's saying then that horizontal gene transfer is sort of the default state. It's the earliest state that you will get in the evolution of cells, and then I will expound the alternative view. So I'll say why I think there are problems with this view, and then give you my unbiased opinions. So in order to do this, why do I think it may be something that has evolved rather than not requiring an explanation? I would look at the ways in which mobile elements, those are genes that can move around, operate, and modern systems of genetic inheritance, and the sorts of things that are likely to be barriers in early genetic systems, things that are going to make it difficult for genes to move freely across. So I will argue then that these modern systems look to me like they're the consequence of evolution, and that they're a product of natural selection, and that there are probably theoretical problems with Wose's model. And that was the motivation for talking with Axel during the summer. There may be some good scope for actually formalizing the models and testing under which conditions they may operate.
Can you maybe, in a nutshell, just explain Lamarckian as opposed to...
I'll do that at the end, actually, because
okay.
that will come as a sort of separate thing. So, this is another quote then from Wose, and I picked it out because it brings me into a couple of concepts. So he says, and the ancestor cannot have been a particular organism, a single organismal lineage. It was communal, a loosely knit, diverse conglomeration of primitive cells that evolved as a unit, and it eventually developed to a stage where it broke into several distinct communities, which in their turn became the three primary lines of descent. So what he's saying then is he thinks that the whole group of organisms were pretty much evolving as a single unit. And for an evolutionary biologist, two things start to come to mind when you read this sort of thing. One is altruism, that is being good unconditionally, if you like, and then what we call the units of selection. And I'll explain both of those terms as I go along. So to start off then, we'll start with a very simple model of early evolution, the simplest conceivable RNA world. So as you've probably heard, the RNA world is this idea that before DNA and proteins evolved, RNA was both a genetic material that carried information, and it was also able to copy that information. And so we can conceptualize that in the following way, that we have some piece of RNA carrying information that enables the production of this thing here, a replicase, an enzyme that's able to copy more RNA so that it can read itself effectively. You produce a copy, and then you can read and produce more copies of that information. So that's the simplest conceivable, I guess the simplest conceivable model that we can have. We don't know if it's realistic, but it's useful for the current purpose here. What we will expect to happen then is that the copying, as this replicase comes back and reads the original template, that copying will not be perfect. We don't have any perfect copying machinery. Our enzymes that do the same thing that copy DNA, they're not perfect copiers. So this thing will make changes. That variation will lead then to competition between these replicases, because they'll all be slightly different versions of one another. And that's the raw material on which evolution can act, or natural selection can act. So the problem, of course, with this type of thing is that there's going to be a limit on how much information can be coded. And to give you then an example, this is an example that I've taken from a paper in Nature from 1979 by John Maynard Smith, and it was actually a critique of an idea, and we'll get onto that in a minute. But the first thing we need to say is, we have a message, in this case it's God Save the Queen, and we want to copy that message. We say it has a certain amount of information, in this case 5 bits of information per letter, so in total here we have 75 bits of information. Now if we suppose that the error rate in copying this is 1 mistake every 50 bits, then we know that trying to copy that is going to lead to the rapid loss of that message, because we can't maintain that much information with this rate of error. So if we replicate this as a single message, God Save the Queen, errors will accumulate, the message will be lost. And this in simple terms is what Manfred Eigen, who was a Nobel Prize-winning chemist, called the error threshold. Eigen's solution to that was to do the following thing: to say we can still maintain that kind of information even if we have this error, one error every 50 bits, by dividing it up into what he calls a hypercycle. So imagine each one of these is one of these little RNAs that can copy itself, like in the picture I showed just a moment ago. So God is an RNA that can copy itself, save is the same, and so on. So then the longest word we have is queen, which is only 25 bits, so that's well below the error threshold. So provided each one of these copies itself, and then copies the next word in the sentence, you can maintain that entire message. So in terms of how an early RNA world might have got more complex, one solution then suggested by Eigen was that this type of mechanism, which he calls a hypercycle, would have emerged. Now the reason I'm showing you this is that this is an example of what we call altruism, because what's happening here is that in order to preserve the entire message, everybody has to cooperate. Okay, and we could imagine, this is what Maynard Smith says, we could imagine if these are RNAs, they're all using the same 4 letters, right? So they're all competing for the same source of letters. So if God suddenly gets a bit better at copying itself than it is at copying save, the result is going to be that it's going to tend to increase, but save will start to decrease because it's getting less help from God here. Okay, so that means that because of that, each word is actually independently being selected. Okay, so the unit of selection is actually each one of these, not the entire cycle. You'll get competition between the words, and the cycle will collapse. Alright? So just then to show that more clearly then, if we imagine that we ended up with a version here, a parasite that receives help, in this case from 2, so it may be a mutant of 3, it receives help from 2, but doesn't give help to the rest of the cycle, and it gets more help from 2 than 3 does, that cycle will crash. So because we're getting variation in these, each one of these is being imperfectly copied, you're going to get mutants arising. If those mutants are able to copy themselves at the expense of the rest of the individuals, then that altruism breaks down. Okay, so there are ways to solve that, and one example of that was that if there is a limitation in the capacity to spread across, so rather than, so if we think about Wose's model, what does he have? He had this altruistic system, and we just pointed out that may be a problem. The other thing he had was what we call perfect mixing. Any gene can swap between any individual. And the interesting thing was that a solution to this dilemma of the hypercycle was proposed in which, even though it's altruistic, it can survive in certain circumstances. And those circumstances turned out to be when there was limited spread. So there wasn't perfect diffusion. There was effectively a flat 2-dimensional surface. So the parasites couldn't invade the entire population. They could only invade a particular segment of that population. So under those circumstances, a hypercycle would be stable. So what that example is telling us then is that these 2 key points of Wose's model, if these points can be generalized to his model, would show that his model is not going to work. Okay, you're not going to have perfect, unabated transfer between any cells in a system because of the spread of parasites that will bring down this very altruistic system. The other thing we know is that, and this is a solution from Maynard Smith and Satinbury, is that selection, as you get better at copying, you will tend to move away from a system that's like this because of this problem. This is exactly the same thing. B is being favoured over A because it's receiving help from A and tending to copy itself. Now the way to solve that, another way to solve that rather than having this limitation to spread, is the genes become linked on a chromosome. So if you have 2 genes together in this way, we'll often define, or several genes, we can often define that as a chromosome. Okay. In fact, sometimes we can talk about a single gene representing a chromosome for viruses, but in this particular case, what we're concerned about is that if they are linked, suddenly what happens is that this advantage of tending to copy oneself is eliminated, because suddenly this whole piece of information needs to be copied all at once. So that ability to for B to copy itself at the expense of A no longer exists. That will reduce the ability for a parasite to spread and bring down eliminate that more complex cycle. Okay. So, another thing that Carl Woese has argued is that horizontal gene transfer is very good because it will generate variation. If you imagine you had a whole load of sets of different genes with different types of characteristics, you could start to mix and match those genes and quite rapidly produce something that was much better than perhaps the starting material. The problem, so this in a sense is equivalent to what seems to happen with sex. When we reproduce, there is the genetic information from two separate individuals comes together, you get a process called recombination, which is like shuffling, and you get different combinations coming out. So while the offspring are still similar to the parents, they carry a combination of the information from the two parents. So one potential benefit of sex is that this shuffling enables advantageous combinations to turn up. A disadvantage potentially is that it could break up very good combinations that already exist. And so there's been a lot of work among theoretical evolutionary biologists on this point. And all I will say about that is that the problem with extremely high rates of horizontal gene transfer, as Carl Woese is describing, i.e. completely dominating is what he said, is that while it will bring good combinations together, it will rip them apart equally as efficiently. So there's no way that they can stay together because they're being ripped to bits because of the shuffling being so high. So then hence the line at the bottom there, too much sex can be a bad thing. So then just to summarize, what we know about early genetic systems, a lot of that is we pretty much learnt by proxy from people who work in things like the telecommunications industry and have to worry about information loss. Okay, and the same sorts of features that were key to producing effective telecommunication systems we see in nature. We have things like multiple copy genomes. So in systems where there's lots of mutations, you get, if you have lots of copies of the information, it's the same as backing up your hard drive. You're less likely to lose the information if you have more than one copy. We also know that you can get some changes in the information without necessarily losing it. So some of the changes will change a function of a gene, and some of them will just be equally as good as the original. So not all the changes are bad, if you like. So there's some redundancy, I suppose you could say. We also know that there are mechanisms of repair and mechanisms to shuffle away and produce good and bad combinations. So we know of recombination. So if these are both mutations that have occurred, and this is one piece of DNA and the other one here, this enzyme here would be, by carrying out this recombination, both of the mutations end up on one strand here, but this one here has gone from carrying a single mutation to now carrying no mutations at all. So we see all those sorts of processes in, for example, viruses that have very high rates of mutation. They make use of all of these types of error correction mechanisms. So all of that is more or less saying that horizontal gene transfer is going to be very bad for early systems that are carrying information. If we then switch to modern horizontal gene transfer, I'm going to look at a number of different things here that I will try and explain. And my starting point will be kind of almost ecological. I'll make the argument that selection seems to favour compact genomes in bacteria and archaea, these simple single-celled organisms. And the reason for that seems to be that replication is, in many cases, the slowest step. So I'll explain that as we go. So here is a picture of your bog standard bacteria, E. coli, growing on a petri dish. This is sped up. Every 20 minutes, if conditions are good, you get a reproductive event, one cell splitting into two. So, and of course that's going to happen exponentially. One goes to 2, 2 to 4, 8, 16, and so on. And so very rapidly, the bacteria can spread across that resource and consume it. In terms of that type of strategy of growing, it's obviously going to be very important that that you can grow very rapidly. And this type of strategy, we call this an R strategy, happens when you have sort of boom and bust cycles. Sometimes there's loads of things to metabolize, and sometimes there's very little in the way of resources. So if you think about bacteria, if they happen to land on a plate of food, it's party time. Once that's disappeared, they have to wait until they arrive at the next resource. In terms of what that means for genes and horizontal gene transfer, we can say that there's obviously selection for fast growth in this type of environment. At the same time, we know that they have a particular way of copying their DNA which is quite slow, and I'm going to make two arguments here. I'm going to argue that their particular form of replication, which I will show you in a minute, the copying of DNA, will favor loss of genes that are not being used, and that this pressure for fast growth has resulted in fast gene expression. I probably want to talk about the second point. So in bacteria, what we have is a situation like this. Here is what we call the origin of replication. This is the chromosome. When you copy this, it basically starts to open the two strands up in this way, and you get copying in two directions, one going that way and one that way. And these two forks will meet somewhere down here in the middle. And if we look at the rates in which that occurs, the rate in which that fork travels is about 1,000 nucleotides a second. So each nucleotide is one of these four letters, A, G, C, or T. And if we compare that to our own cells, our replication runs about tenfold slower than that, somewhere between 50 to 100 nucleotides a second. The difference is that we have much more DNA, so it ought to take a lot longer to copy. The difference is that we have many of these ories stretched along our DNA, so each one of them doesn't have to go very fast. So this is copying in parallel with many positions, as opposed to a biparallel replication with only a single origin. The details here then, just all you need to know really is that replication overall in something like E. coli takes on the order of an hour, and yet this fast growth in optimal conditions enables division every 20 minutes. So that means it takes longer to copy the DNA than it does for the cells to split in half, into two daughters. We see the opposite actually in our own branch of the tree. So we can see an example of the fruit fly in the early stages of dividing a fertilized egg. The time it takes to copy the genome, which is orders of magnitude larger than bacteria, is about 3 minutes because of this replication in parallel. And the time it takes to divide, in this case the nuclei which are carrying the DNA, is 9 minutes. So here replication is not rate limiting, here it is rate limiting. We see the same thing more or less in archaea. So there are a couple of different examples. I just run you through this same example, this one example just to be quick. Here we have a single origin again, quite a fast polymerase. It's about 1/3 of the rate of the E. coli one. It has quite a small genome, and it ought to take about 49 minutes to replicate. I'm not sure what the division time is here. I think in two of these cases replication is rate limiting, and in one of them it isn't. In this case here you have sort of an intermediate. We have in both of these cases 3 origins of replication. The polymerase is about 3 times slower, and the expected time for replicating is on the order of an hour. So in these cases it looks like we're getting the same picture as with the well-studied bacteria, that the slowest thing is copying the DNA.
Are these experimentally measured replication times?
Yeah, those are all from experimental data. Not my experiment about other people's. Okay, so in terms of, if we look at then at the genomes, it looks like they don't waste any space. So this is a plot showing the size of the genomes, okay, the length of the DNA for a particular species of either archaea or bacteria, and then the number of genes. And there's a clear correlation here. So there's no cases really where this deviates. Whereas if we look in eukaryotes such as ourselves, we have huge amounts of DNA and very few genes. So the only examples that are different to this are guys like this here, and this bacterium here we know has lost a large number of genes very, very recently in adapting to a lifestyle as a parasite. So there we can see that it's in the process of degrading large numbers of genes. But the others, they all pretty much, regardless of whether they're archaea or bacteria, fit along this line very surprisingly neatly. So an additional thing that's been established then about bacterial genomes, as well as being compact, they don't seem to waste space, they seem to turn over very rapidly. And this is where the horizontal gene transfer comes in. We know that all cells require some core set of genes in order to run. They need to be able to copy DNA and produce proteins, for example. They need to be able to make a membrane. There's going to be a few sets of genes that are absolutely essential to the running of any type of cell. And then there are genes that have been described as being flexible. They're maintained sporadically in terms of the individual. You'll find closely related bacteria, for example, that carry things like antibiotic resistance in one strain and another strain lacks it. So these genes, for a range of different types of bacterial activities, are either present or absent, and the only way we can explain that is that these genes are turned over rapidly. They're either, if they're in the genome, they will be maintained, presumably because they're required, and that they can rapidly be lost from that genome if they're no longer being used. And we can explain that in two senses. One is that there may be actual selection pressure to remove them, given that very clear correlation between how compact the genomes are and the number of genes. But there may, it's also true that if something is not being used in evolution, it's rapidly going to, if a mutation occurs that ruins a particular gene, it's not going to matter a jot to an organism that's not using that. So it could be easily the case that one loses a gene. So we could expect degradation of the information rapidly, but to account for loss as well, there must be an additional selection, and that may be then this requirement to quickly copy the DNA under certain conditions. So it's also true then, to answer a question that came up earlier, that rather than single genes, you may even get whole, what we call genomic islands, carrying a whole suite of genes, for example, that enable infection of particular types of hosts. So there are genes carried on plasmids that I think things like the ability to cause these horrible stomach ulcers, that's carried in some species of this bacterium Helicobacter carry that, others do not, and they're not harmful. And that's something that's transferred within a population. So you can get this process then of receiving some genetic information. Those genes can be inserted into the genome, so you have now extra genes, and if those genes are doing something useful, they will be maintained. If not, they may be lost again. Okay. So if we think about that from the point of view of the organism, it's probably a very good thing to be able to dump the genes and to be able to pick up nutrients. But why are the genes being transferred in the first place? Now this is where we will sort of start to set up why the Lamarckian idea is not good. If we think about it from the point of view of the organism, as it were, to talk in anthropomorphic terms, that's a great system. You just pick up what you need and dump what you don't need. It's like having a backpack. You just put the things you need into it and then leave the rest of the stuff or throw things out as you no longer need them. Now, if we think about it from the point of view of genes, there is a risk that a gene can go to extinction by this process. If it's not being used and is lost, it can, and there is regular loss, it may just completely disappear from the face of the planet. Genes that are able to enter into bacteria and be retained will tend to be favored during evolution. So that means that they either have to be doing something useful or doing something tricky to ensure that they're retained, and they also need to have some mechanism by which they will be taken up by a passing cell, if you like. Okay, so there we have two different sets of selection. So just to summarize what I was talking about with the genomic islands, we can have these genomic islands inserted. They may turn out to be useful either to adapting to a particular environment or becoming a pathogen of a particular organism and so on. But they will only be maintained if they actually, I guess you could pick up the right genes at the right time, to say it in simple terms. So those genes could easily be integrated and lost. They will only be retained if there is selection to retain them. Okay, so what you may have heard is, of course, the human genome is littered with DNA that doesn't seem to be required to make proteins. And I'm going to say this very briefly. What we know is that one of the reasons for that is that in an environment where there is sexual reproduction, as in ourselves, these types of elements, these are selfish elements that do nothing other than jump between genomes but don't confer any advantage on their host. They, under sexual reproduction, they can rapidly spread between individuals. And to give you a quick analogy, it's sort of like an STD. It can easily spread between individuals because you can have an infected and uninfected individual reproducing, and the offspring will receive this particular gene. So it can spread, and I won't say more about that. In the case of an asexual system, such as, so it's a clonal copying, which is what you may see in, for example, bacteria, a gene that is not conferring any advantage, it's selfish, if you like, it costs the bacterium something to be there. As it starts to make additional copies of itself, if those copies confer a further cost on the cells, they will start to become sicker and sicker relative to individuals who do not carry them. So the result will be that these things are likely to go extinct in this type of environment. They will flourish in a sexual environment, but they will either cause the individuals in which they've infected to die out, or they could potentially wipe out an entire species, simply because they're no longer, they're not being constrained, they've jumped in, and everybody dies because they've got sicker. We think about that in terms of horizontal gene transfer, it's going to be much the same thing. You get entry of a particular gene, it jumps somewhere, and anybody who picks those up is eventually going to suffer from this additional load and will presumably go extinct. So you expect then, from the point of view of the genes, this is a very bad strategy. What do they do about that? We expect these kinds of elements that can jump around in bacteria, because bacteria are primarily clonal, they reproduce this way, not sexually. They will either be cautious in the way that they spread, or they will be beneficial. That is, they're saying, when they come into a host, they're sort of saying, hey, I can help you with this particular thing. Right? I'm going to skip that. So we see an example of that cautiousness here. There are two strategies in which these things called transposons, that just means jumping genes in genetic speak, can operate. They can either jump in this conservative manner, which means cut and paste. So they cut out of one particular point in a stretch of DNA and insert into another piece. So the outcome is that they've moved, but the number of copies is still the same. In this case, it may be one. There is another method and another mechanism, and that's replicative, where an additional copy is made so that you started with one, you now end up with two. And there's a bit of a blur there, as the biologists here will know, but I won't talk about that. What I will talk about is that what we tend to see is that these bacterial transposons, these jumping genes in bacteria, tend to have this strategy, whereas the ones we find in, for example, our own genome, they're almost exclusively this strategy here. And we can see that in a few cases where they've been studied in bacteria, they will tend to behave like this. So less than 0.1% of the cases are of that type of copying. But they will, if the bacterium is under stress, they sometimes will switch from that to that mechanism there, which may be an adaptation to trying to move out and find somewhere new to live. Okay. So in addition to genes that may be useful under certain conditions, and that's their meal ticket for staying in a genome, we have genes that use devious ways and avoid being lost. So they're effectively behaving like a protection racket, and I'll explain how they do that. So one particular favorite of mine is these systems called restriction modification systems. And basically what they do, what we originally thought was happening was this was a form of immunity that bacteria carried. In the same way as we have immune systems, it looked like bacteria had immune systems. They carried these extra pieces of DNA here, which we call plasmids. And on one of these plasmids there were two genes. One is a modifier, we call a methylase, and what that does is that tags the DNA here in different positions, and that tag is sort of like a form of protection. It also, the other gene here, the R, produces these little enzymes here that chop up DNA. So all they do is they cut DNA. And in the absence of those tags, this DNA would get cleaved. But because the tags are there, this DNA is safe, and these guys cannot cut up the DNA. What they can do is if there is an invading, for example, a viral DNA that doesn't have these tags, they will cut that out. So that looks like a form of self-defense. That may be the case, but it turns out that there is a price to be paid there. So here we have one of these cells exactly the same. Now, if that cell by chance loses this plasmid carrying these two genes, the outcome is death. And the reason for that is that as the cell divides, it no longer has a way to continue to tag itself and keep itself safe from these little hungry enzymes. And eventually they will, you just need one of these to be missing to start getting a nick. So as those disappear, these guys will stay within the cell. There'll be a fewer number of them, but they're sufficient to cut up that DNA. So this is what's been described an addiction mechanism, because once you've received one of these plasmids, you can't get rid of it. The reason for that is that the cells will die out in the population. So they can spread because these plasmids are mobile, they can hop between cells. But once they're there, you know, they're like the mafia in the movies. You're not going to get rid of them from your neighborhood. It turns out that they also have a very good system of territorial defense. So there's this phenomenon called plasmid incompatibility in bacteria, where you see this case, in this case I've shown it as a new plasmid has come in and displaced that particular plasmid. And the outcome is exactly the same as this case here. You no longer have the genes to produce the antidote, and this happens. So that means that it's also a good system of preventing competing plasmids from coming into this population. So it's this defense, in a sense, is a side effect of territorialism at the genetic level. So these guys are, in a sense, they're not useful, particularly useful. And a good example of that is, while you could always argue that that looks like a good thing, you can have 2 of these, or 3 of these, or 4 of these, or 5 of these. You know, so it'd be the equivalent of some poor restaurateur with 5 different gangs all saying, look, pay us money, and we'll look after you, you won't get, you know, there'll be no trouble, that sort of thing. So they can rapidly be a genetic burden. Okay, so this is a system where you can get around this problem of being lost. So if I sum that up and say what I think that means, all of those systems indicate to me, actually, I'll take the second bit first. Those don't look like throwbacks to some early system that don't require explanation. They look like the product of natural selection to me. So we see natural selection on the size of the DNA in bacteria and archaea. There's constraints on the speed at which the genetic information can be copied, and that's heavily linked to their lifestyle, that they're very fast growers under good conditions. It seems that there are clear strategies employed by certain selfish genes to prevent themselves from being lost. They also prevent cases where they will be so damaging to their host that they disappear themselves. A number of these, if you like, helpful functions survive because they're only partially helpful and tend to get lost. They only survive because they are mobile. So all of those things look to me, rather than being something ancient that doesn't require an explanation, they're all adaptations that the genes have taken on, if you like, to be able to survive a particular genetic environment. So in terms of what that means for horizontal gene transfer early, it may have been there, but one, these modern examples are not the type that we would see. And the second thing is that, at least from looking at other similar systems, it seems that two of the problems with the model that Carl Woese has proposed is that it doesn't take parasites into consideration. It would appear that a system such as his would be crashed by parasites, and while these beneficial combinations could be made, they could equally be ripped apart. So it doesn't seem to me to be a Darwinian system at all that's going to work. You're not going to get new favourable forms of organisms that are going to do well without sort of a barrier to transfer. This isn't going to operate. So then the final couple of slides then I'm going to address this last point about whether horizontal gene transfer is Lamarckian. So this is a quote I've taken out of this recent essay from Goldenfeld and Woese from this year. I should have got quotes around that, but they've argued then that early life evolved in a Lamarckian way. As I showed you before, they said that they thought the vertical descent really wasn't very important, and it was much more horizontal gene transfer. As I've already pointed out, they were saying that transfer would be such a great way of producing genetic novelty by producing new combinations. And they end with this point here. They say, thus we regard as regrettable the conventional concatenation of Darwin's name with evolution, because other modalities must also be considered. So they're saying, really, we think it's Lamarckian, and Darwin's model, really, it doesn't have anything to do with this. And I got very upset about this. So let me explain to you what Lamarck said, so that I can explain to you what I'm arguing against. So Jean-Baptiste Lamarck, he didn't actually come up with this idea. This is a very old idea, the inheritance of acquired characteristics. But he developed the idea. It wasn't even the main idea in his actual thesis, but that's the one he's been remembered for. He sort of thought, kind of take it or leave it. So the guy's been a bit maligned by history. But I guess the classic example of how this operates then is the giraffe's neck. So if you imagine you had these short giraffes here once upon a time that were stretching out to try and reach leaves up on the high branches of trees. Over the course of their lifetime there would be stress on their muscles and so on, and they would have an effect on perhaps the length of the neck. It might get slightly longer. That change during the life of the giraffe would then be inherited by the next generation. Okay, so they're talking about a direct inheritance of a character, the length of the neck, that's been acquired during the life of that organism by trying to do a particular thing. Okay. So in the modern form, that would be of the following type. So I guess you're familiar with this, what people call the central dogma, the transfer of information from a gene in the form of DNA to a working RNA copy to then the final product, the protein. So here we have the genotype. This is then a working copy, and this is what we call the phenotype. So the character of that particular gene. Now, in order for, it's been pointed out that in order for Lamarckian inheritance to work in the molecular sense, what you would need is something like this reverse translation. So this kind of copying, which we see, that wouldn't constitute a Lamarckian form of inheritance, because all this is a copy of that. It's nothing to do with a characteristic. It's just sort of this is the characteristic here. This is the equivalent of the giraffe's neck. This is just the messenger. Okay. So what we would have to happen is that the protein, which is the equivalent of the neck, would undergo some change, and that change then gets transferred back into the DNA. Okay. So that is how Lamarckian inheritance could work if we observed it in modern biological systems. There's no reason why that shouldn't happen, but we haven't observed any cases of that. So if we contrast Lamarck's idea with the modern version of Darwinian evolution, which we call neo-Darwinian, there were two key aspects of Darwin's original model that were updated in the last, with ongoing research. So the first was descent with modification. So that is then that offspring resemble their parents but are not identical. When it became neo-Darwinian, when we realized how that happens, and that was the form of inheritance that the Austrian monk Gregor Mendel described. So there is variation being generated by mutational changes in the genes. So the sequence in a gene will change, and that variation can then lead to heritable differences. So different individuals in the population will carry slightly different versions of those genes. And that difference is what natural selection can operate on. Those individuals then that are best adapted to their environment will leave the greatest numbers of offspring. So in the case of giraffes, how that works then would be that there would be variation in the genes affecting the height or the length of the neck, and that if having a longer neck is a good thing, then those with the longer necks would be fitter. They would tend to have greater access to the resources and therefore leave more offspring. So then the neck over time in that species would elongate. Okay, so when Woese is saying horizontal transfer is Lamarckian, if you look at it superficially, it looks for all the world like a Lamarckian system. You have bugs behaving this way, normal vertical inheritance, but you also have transfer of a trait in the form of a gene that carries out something like antibiotic resistance from one individual who can be unrelated to another. Okay, so that looks like the equivalent of receiving a longer neck during your lifetime. So I say that it is not, and the questions we have to ask are as follows. One is, how is that variation being generated, and is it the case that natural selection is operating? Natural selection could operate with Lamarckian inheritance, but I think if we forget about the organism, stop focusing on the cell, we can solve this and get a much clearer picture. So when we think about these genes, and we can even go back to the tree, they're still evolving in a Darwinian manner. You're getting changes occurring because they're copied, and small changes are appearing in those versions. So there will be different genes that perhaps may be slightly better or slightly worse at being transferred, for instance. Some may be better at transferring themselves, some may be worse. That variation then is being acted upon by natural selection, so that's DNA changes, natural selection acting upon that, and we can in fact describe that using a tree. So that was in fact one of the key points that Darwin himself worked out, was that his model would lead to lineages splitting in this manner here. So even if we take genes that are transferring like crazy, we can still look at the similarities and differences and produce trees of this type here. So it doesn't really matter that the genes are not staying with the organisms. What we're seeing is that the genomes are in flux. Genes are coming and going. Some of them may stay there. but some of them are not. What's happening really is that there are bonds of cooperation between genes or groups of genes, and those are being formed or broken. So if there are antibiotics in the environment, antibiotic resistance genes will cooperate with the rest of the genome. If those antibiotics disappear, so will those antibiotic resistance genes. None of the changes that are occurring here are not genetic. None of them are cases of this reverse translation, changes from the protein and then being inherited back into the DNA. So that, again, it looks really like the mechanism of transfer here is a gene survival strategy that's the product of Darwinian evolution. It's not Lamarckian, and it does require an explanation. So if I then, my final slide, say a few concluding remarks about the tree. While certainly there's been panic about horizontal transfer and what it will do to the tree, the thing is that it may not necessarily invalidate using trees to examine the relationships between organisms. Okay? There's no requirement that all the genes have been inherited vertically. So if some of them have not been subject to transfer, then we can use those to look at the evolutionary relationships between species of bacteria. For those genes that have been transferred, they will instead reveal this history of cooperation with other genomes, other assemblies of genes. And so we can trace them moving around. So it's saying to us, well, yes, it's more complex, but we don't need to throw everything out as a consequence of that. Okay, I'll stop now because I'm sure there are lots of people wanting a drink. So thanks very much for listening.
Thank you. And questions?
So if we think about your middle statement here, those genes that have stayed on sort of and been inherited vertically all the time, would those correspond to those 50 or 60 that you had on your first?
Yeah. And the question of whether you can reliably use those to rebuild an actual tree of life, that's another thing altogether, but it means that there may be still some information there.
So have people tried that a lot? Because I mean, the trees we are usually looking at, those are from the ribosomal RNAs.
Yes.
Have people looked at a few of those that are
50
or 60 and at the same time?
Yeah, so one study that was published 3 or 4 years ago now was, Norman Pace was one of the guys on that paper, and they basically took the tree, those genes that gave trees similar to the ribosomal RNA tree, and they found that there were 50 or 60, and of course predictably they were involved in the ribosome, so ribosomal proteins for the most part. But so there seems to be a core of genes that are giving more or less the same signal. While whether everybody agrees that they're particular trees is another matter, since everybody likes to argue over the trees. I think the really good point about that paper was saying, hey, yes, there is this core, and it seems that there is a good percentage of them giving more or less the same signal, at least in our analysis. So that suggests that you can rescue a tree. Yeah.
Is that how you differentiate between those that has been invented vertically and those not been vertically? For example, that you can build this tree or...
Yeah, it gets complicated. In principle, that's one of the good ways to do it. But it can kind of evolve into debate as to whose tree is correct. I mean, you need to have a reference tree and say, we agree that this tree is correct, and therefore if we get a different tree from this, one of the possible explanations is that gene is being transferred about. Another thing may be that, well, that gene just isn't very good for building trees, and the debate at the time was then, well, actually I don't think yours is that good for building trees either. So there was this haggling over whose molecule was the best one to build trees from, and probably the answer to that is that no one gene is going to be sufficient for building a tree that spans several billion years of evolution. There's not enough information on that sequences. Yeah.
So this resistivity problem in bacteria against antibiotics is actually solvable
Yes,
If we stop using certain antibiotics, then 50 years from now we can start using them.
Yeah, that argument has been made by people working in that area that the biggest problem is that because we continue to use them, the resistance will build up and spread. And so...
But this is very different from, let's say, more complex organisms then, because I've also read that, as an example, I think it was like, whatever, the Nordic people here are more resistant to AIDS because of some previous epidemic that happened thousands of years ago, and this is still in the
right. This particular, there it's just a particular version of a gene that's what we call an allele. So it's to say everybody carries that gene, but the version of that gene is different among Nordic people.
But there's a memory there thousands of years.
Yeah, that bit's the bit that people are debating. What exactly? How come that gene that allele is there? Is it? One of the points that was made, I think, the idea that was put forth in the literature was that was to do with the bubonic plague, and this is a pre-adaptation that turned out to be good for HIV as well. But that bit is more controversial. But in that case, it's for some lucky reason, there's a group of people that carry a particular variant of a gene that everybody carries that means that they have a better chance against this particular pathogen. So that's different from the case of some bacteria carrying antibiotic resistance and some not, because it's not the case that they all have the gene. Some of them actually have the gene, some of them don't. But perhaps in terms of the outcome, it may be somewhat similar. Some of them survive. Yeah.
I try to understand the bottom line here is that this horizontal gene transfer is like, what was the term, Lamarckian? Lamarckian. That was claimed in this article in Nature, and you don't like that.
I don't.
And so how do you win such an argument then if you sort of go into debate? I mean, because you cannot identify critical experiments to do, or you cannot.
Well, there have been plenty of critical experiments done in the past. I think the famous thing was Weizmann, who was the first to really attack the idea around the turn of the century. And he went off and said, look, this is nonsense, and I'll do an experiment to show that it's nonsense. And that experiment was to cut the tails off of mice and show that the next generation had mice, which perhaps these days might not be considered so ethical. In the case of this, what he's claiming is interpretation of already known facts. We know this occurs, and we know what is required to explain. I guess it's already been worked out what would constitute a Lamarckian model of evolution, and that all that's really happening is falling into the trap of thinking of the cell as somehow an unchangeable thing, and because you're getting transfers, that's an acquisition of, and the inheritance is therefore fundamentally different. However, if you ignore the cell and think about the genes, everything about the function of the genes, the way in which they're able to spread and so on, can all be explained by Darwinian selection. So there's really, it ends up being the only really, I guess the way I would solve it is a polemic, just go out and grumble and complain. There's no experiment needs to be done because all the information required to resolve that debate is already known to us. So if he could show then that there was something like this happening, then I think people would say, oh, that's fair enough. And that would be the resolution. That would be proof of a form of Lamarckian inheritance. And you were getting changes after the proteins were made, and that those changes were then being inherited, and the next generation was being affected. So, I mean, these kind of statements about Lamarckian inheritance come up in the evolution literature quite frequently, and there's a long history of them being shut down because of debate, really, without requiring any more cutting of tails of mice.
has there been any response? I mean, has there been like, this was published in Nature?
Not yet.
Reply or something.
I wrote, but they didn't want it. So, so I'm going to write something up about that. But you know, the problem with these essays that have now got popular in Nature, you know, is that they invariably provoke a lot of responses. So you know that they have to field a lot of correspondence as a consequence of these. Most of them won't get published. So there's a lot of controversial points that are being put forth in these essays. I think the vast majority of them are on biological topics, and there's a lot of debate surrounding those essays, but the right of reply is very limited in that particular format. So I will write this up.
I'm also supervisor always say Nature is like a newspaper.
Yeah.
So you shouldn't really, you know, it's like the expression of science.
Yeah. News of the world.
Yeah, I don't quite understand how the gene gets from one cell to the other.
Oh yeah, I guess I didn't actually focus on the biology particularly. It'll take another hour. So one of the things that you see, and there are lots of different ways. You can do a virus can infect, and I should have showed you this picture, a schematic of a virus that looked like this. And the virus can send its DNA through this structure that allows it to enter the cell. And so you can get a virus infection is one way. Another is that on a lot of these plasmids, these extra pieces of DNA, they carry genes that enable the formation of a structure that we call a pillar. And that structure enables DNA to pass through between two individuals. There are other mechanisms. It seems that some bacteria are naturally just able to pick up DNA from their environment. I don't think the mechanism there is particularly clear. For some reason they seem to be very good at it, but how they're doing that mechanistically is clear. I don't know if you know it. No. So there seems to be lots of ways to get in, but there are also barriers. So, but this is probably the classic textbook example of bacterial sex.
A follow-up question.
Yeah.
From you, I understand that this horizontal gene transfer is a property of the gene and not the organism itself.
Yes, I think that it's turned out to be a good thing for the organisms. The problem with evolution, as opposed to say the way we humans behave, is that if I decided I need to put different things in my backpack every day, I'm not going to say, well, I don't need my knife today, I throw it away. I'm going to keep it and I throw it around so the next time I need to fight off a bear or whatever it happens to be that I'm doing with my knife. Evolution doesn't have that kind of foresight. If something is not immediately useful, there is a chance that because of this process of copying being imperfect, you get mutations and that information can be lost. Unless there is a reason, a selection pressure to maintain that information, it will disappear from a population. So selection is required to maintain gene functions, and so genes that are only required occasionally, something like, say, occasional need for antibiotic resistance, the problem is going to be then that those genes can rapidly be lost. That's bad in the long term for the organisms, but in the short term that may actually be bad to hang on to them, and that we perhaps get an indication of that looking at how long it takes them to copy their DNA relative to how quickly they divide. So there may be in the short term a benefit to losing that information, not just degrading it but actually throwing it away. And it doesn't really matter that in 100 generations it's going to be jolly handy because someone's just going to have popped a couple of tablets of penicillin. That's not going to matter a jot in terms of how evolution occurs because it can't stockpile for a rainy day. Of course, from the point of view of the genes, the outcome will either be you either go extinct, or if the gene has a mechanism by which to continue to move about, it may survive. So the only genes among those that tend to be lost that will survive will be those that have developed mechanisms to enable them to be transferred. So they can still potentially go extinct, but that will reduce the chances of them going extinct. And an additional thing, of course, is how easily they can transfer. So you may have heard, of course, that there needs to be transfers all over the tree of life, and that you can get genes from bacteria moving into archaea that are separated by billions of years of evolution, probably. And one thinks about it in terms of gene strategies. Those genes that, if there's high rates of loss that can spread most universally, they will be the ones that tend to survive. So we may therefore expect that gene transfer will be across very broad distances, because there will be potentially an advantage. If one can only stick to a very small range of a very small isolated population of bacteria that are very similar, the chances of loss may be much higher than if one can invade any number of different species in the community.
This may be a specialist question, but actually the last picture you had up here with Lamarckian
That one?
inheritance, right? No. No, I think it was where that
one?
That one, yeah. So the chromatin structure of DNA can be inherited. So where would you put that in this?
Are you thinking of like epigenetics?
Right.
Well, I don't think anyone's shown long-term sustained inheritance. I mean, for example, with epigenetic signals, they get wiped out very rapidly. So there have been studies, for example, so epigenetics means generally tags that are on the top of the DNA, additional information saying, for example, switch off a gene, switch on a gene, that kind of information. And some of those affect characters, such as the example I'm thinking of is coat color in mice. And so it turns out that whether they're white or brown or something in between is determined by these markers on top of the DNA rather than the actual DNA itself. And a group in Australia were actually they tried to select for a particular colour among the mice. So normally if we were animal breeders, we'd say, oh, we like brown mice, so we kill off all the white mice and just keep the brown ones, and then hopefully over a few generations we will have brown mice, and then we can make our millions selling brown mice to everybody. Now, in this case, they were trying to do that, and they would apparently be selecting for brown mice, or what, I can't remember if it was brown and white, but one of the colours. And then after a few generations, you just got the original spread of colours turning up again. So that while there is clearly epigenetic inheritance across a generation, it doesn't seem to result in long-term genetic changes. There's no reason why it shouldn't. There may, but we haven't seen any clear examples. There are possible mechanisms.
But if it would stay over several generations, where would you put it in the scheme here?
It still would not be affecting inheritance, because it can't be acted upon by selection. So there's this artificial selection thing would be the equivalent of saying, well, if you're a brown mouse, it's really good, and if you're a white mouse, you get picked up by the cats. But it didn't matter, because you end up with white mice all over again, simply because it's an epigenetic trait, and therefore it doesn't matter how effective the cats are at knocking off the white mice, they still reappear. So unless that one possible mechanism is that you get the silencing via methylations, and those methyl sites are prone to being have higher rates of mutation. So if that mutation also corresponds with a constitutive silencing of that gene, that might be a case where you'd have perhaps something you could consider as monarchic. It's hard to say. There might be some complications with that, so I wouldn't put my money on it. I haven't thought it through.
Because otherwise, I guess the common notion for anyone would be that what you have drawn here as the Lamarckian pathway would be impossible. But so if one starts thinking along the line of epigenetics, that's not
No, and that's
Not
one thing.
completely out of question
Absolutely.
knowing chemistry and biology.
No. So there's two. I mean, it's been recognised for a while now that while Lamarckian inheritance was disfavoured, it's a scientific theory, and there are conditions under which it may be fulfilled as a mechanism of inheritance. There's no reason why it can't occur, but I think it was maybe Monod had said that, well, the equivalent principle happened, but we don't observe it, so it's not that it can't, it's just that it doesn't. So maybe this with this flow of information. So absolutely no reason why it couldn't appear and it wouldn't be included as a proper scientific theory, which is of course why lots of people keep coming back with new ideas. Oh, look, we think this is Lamarckian, but often, well, in every case that I know of, that argument has crumbled on cross inspection, and that's the case also, I think, in this one here.
You explained that the hypercycle model is unstable to parasites, and that this, on the other hand, can be stabilized in the presence of mixing.
Imperfect mixing.
Perfect mixing. Now, mixing, then you also said, of course, would imply horizontal gene transfer.
Yeah, I'm sorry, I meant imperfect, like not perfect mixing, not complete mixing. So that if they're spreading out on a two-dimensional surface, and you get, say, a parasite emerging at a particular point, and you have outward growth, it might be able to grow and radiate that way,
Yes.
but it can't take over the whole.
But that's
system.
imperfect mixing, right?
That's imperfect.
And isn't that exactly what actually saves the parasite instability? The imperfect mixing?
Yes, that means, yes, exactly. So even though that system is altruistic, there are conditions in which it can operate.
And are you then saying that that is actually not what horizontal gene transfer?
Well, horizontal. No, I'm saying that Carl was this model because he says that this complete complete mixing with no barriers looks to me like if you tried to formalize the model, you would run into the same problems as were identified with this hypercycle model. That you will get parasite invasion. I think it's the same general type of problem.
Yeah,
it hasn't been
I see. Because it would imply perfect mixing.
Yeah.
Yeah, I see.
So that's why I'm at least initially. I don't think it's impossible that you would have very high rates of horizontal gene transfer. But in order to argue that, one has to start actually formalizing the model and testing the conditions in which it can operate.
Yeah, right. All right. If there are no further questions, then let's thank Antje again.